Hi guys, Sanket here. Welcome to another session on protein-protein interaction network. Today we'll be looking at the last session of PPIN. Okay, that is your topological protein-protein interaction network analysis. Okay, um, now we have seen a lot of protein-protein interaction networks, some very simple clustered ones. Okay, uh, if I tell you that help me analyze these structures, what is there in front of you? You will be like, oh God, what is this? Okay, so these are very dense structures of protein-protein interaction network. I think one is of malaria and second is of another disease. If I tell you that help me analyze each and every protein which is there in this, help me analyze each and every drug which is there, it becomes really, really difficult. Okay. That is the time we use topological PPIN or topological protein-protein interaction network analysis. What topological analysis does, it divides that entire protein into small substructures. Okay, and dividing it into substructures, it helps us to find those entities. Now I'm saying entities. Okay, entities are basically your nodes, which can be proteins or genes. I can always say proteins and genes because that's not everything which is there in the node. Okay, you can have a lot of things. So I'll say entities. So what are these entities? Okay, which are present, or what are the nodes which are of biological significance? Okay, and that is what topological PPIN analysis helps us. Now there are two main analysis which a network does. There is centrality analysis, and second is your clustering analysis. Okay, your clustering analysis includes some further parts called as motives or shortest paths and all. Yes, centrality analysis has two more parts of it. Okay, so we will be looking at both of these analysis. Okay, of topological analysis. One is your centrality analysis, and second is clustering analysis. Okay, now what is your centrality analysis? Now, by the way, I think looking at this network, you will get to know what this network is, which database we have taken it from. This we have taken it from a protein-protein interaction database called as KEGG. Okay, so KEGG is one of a protein-protein interaction database. There is others. There's String. Okay, there is Mint. So what if I tell you? Okay, help me find in this entire network which is the most important target protein. ये अच्छा, by the way, this network is in cancer protein network or cancer pathway. Okay, अब इस cancer pathway में अगर मैं पूछा कि इसमें मुझे सबसे important protein बताओ. Okay, या मैं तुमको पूछूं मुझे सबसे important gene बताओ कौन सा है. तो ये ढूंढने के लिए, और in order to find this you will need to carry out this centrality analysis. Okay, what centrality analysis does is it basically generates a network and it generates hubs. So what you see, these red nodes, okay, they are hubs. What is hub meaning? If you split this word centrality analysis, centrality को अगर आप break करोगे तो उसमें एक word आएगा central. हम central उसमें डालते हैं जब when you have lot of things connecting to each other. Like if you see in Mumbai, there is a place called as Mumbai Central. Okay, the reason why it's called as a central is because all the trains from each and every place, okay, from all over India, they go and they stop at that Mumbai Central point. That is the central point, what we call it. Is another name what we call as junction also for it. Okay, so that is the junction point, that is the central point. And because it is the central point, all your networks are connected to it. So your nodes, okay, this red color hubs is your central region. So that's why if you can see it's given here, it has a high centrality value. And if we get to know which are those nodes which has a good or high centrality value, इसका मतलब क्या? We will get to know if we feel that one node has a high centrality value, मतलब काफी सारी चीजें उसको connect हो रही है. Okay, and if that node is very, very important, that means it definitely must have some sort of a biological significance. Yeah, so answering your first question was, if that network, whatever the cancer pathway we showed, okay, if that cancer pathway, which is the most important protein, which is the important drug, is we need to find those proteins or those drugs in the network which has a high centrality value. Hope you understood this. Yeah, so your centrality analysis can be measured using different criteria. So there are three basically three important criteria for your centrality analysis. One is your degree of nodes. Now, I don't think I need to go into more into degree of nodes. We have already discussed this very much in your prior sessions. It means more the connectivity, that much will be the degree. Second is global centrality. Now, global centrality, what it means is how your protein-protein interactions, or how that's at a network level. So, your any protein, your any hub protein or any other protein, how is it connected? What is the central connections with the entire network? What is this? That is called as global centrality. Okay, so you have degree of nodes, global centrality. Random walks, as it suggests, is how many steps, okay, do a node needs to take, okay, in order to go from one network to another network. Something very similar to your 6 degree of separation, okay, or your small world effect, that how much one needs to walk, okay, in order to cross from one network to another network, which actually shows how good the connectivity is. So these are your three important the criteria or different metrics which are measured when we are analyzing centrality. Is the degree of nodes is measured, the global centrality is measured, and the random walks. These are three things which is measured. Now, centrality analysis has two. There is one which we call as closeness centrality. Okay. Now, closeness centrality, what it means is, now if you look at this, okay, there is a what is that? एक इक्वेशन आता है, ओके, और सबसे पहला ख्याल जब आता है ना इक्वेशन को देख के, वो बोलते हैं ओ शिट, ओके, अभी इक्वेशन याद रखना पड़ेगा क्या? डोंट वरी, जब चीज कोई डिफिकल्ट लगती हो ना, तो सबसे पहले उसको कवर कर दो। ऑलराइट, नाउ हम उस चीज पे फोकस करेंगे जो हमको सिंपल लग रही है। ओके, अभी ये एक नेटवर्क है ABCDE। अब आपने डिग्री तो किया होगा, यू रिमेंबर द डिग्री हाउ इट वर्क, ओके। इसका थोड़ा सा फंडा डिफरेंट है, ओके। इसका फंडा ये है, दिस इज द सेम मैट्रिक्स, ओके, वी आर, जैसे हमने डिग्री के लिए जो मैट्रिक्स जनरेट किया था, वैसे हम इसके लिए भी मैट्रिक्स जनरेट करेंगे, ओके। सो ABCDE इज योर मैट्रिक्स फॉर दिस, एंड देन अगेन ABCD इज अनदर मैट्रिक्स फॉर दिस, ऑलराइट। सेम हमने वैसे एडजेंसी मैट्रिक्स जो बोलते हैं उसको हमने जनरेट किया। अभी इसके लिए हमने जो डिग्री के लिए कैसे किया था, वी हैड गिवन लाइक A has 2 or something, like A with B it is 1, with A it has C it is 1, okay, वैसा हमने किया था। इसमें थोड़ा सा डिफरेंट होता है, बिकॉज यहाँ पे what it actually looks is how your specific node, okay, how much your node is far away from other node. That's why if you see this term is called as farness of the nodes, like how far it is. I'll explain you, काफी सिंपल है, okay. So हम पहला वाले लेते हैं, okay. So ABCDE, so A with A 0, A with B 1, A with C 1. Now D and E, if you would have seen normal जो अपने पहले के केसेस थे ना, वहां पे it would have come 0 and 0, okay. That was in your undirected edge वाला था ना ग्राफ थियरी का, वहां पे this would have come 0 or 0, but this we are looking at closeness centrality. So we are actually want to see how much something is close or far away. So B and C are very close by, that's why it's 1 and 1. D and E is far away, okay, are you understanding? It's far away. अच्छा, far है तो how much far it is? It is 1 and 2, that is ABDP. So it is 1 and 2, that's why 2. So D is 2 times, so that is the numerical value, it is 2 which is far away, ठीक है, समझ में? अभी E का देखते हैं, E कितना दूर है? 1 2, सर 1 2 3, नहीं इतना लंबा क्यों जाने का, सीधा सीधा जा रहा है ना तुमको, ओके तो इतना लंबा नहीं जाने का, ओके सीधा सीधा अगर आपको मिल रहा है तो ठीक है। सो E का आएगा 1 and 2, ओके दैट्स वाई 2, सो A and E is again 2। B का देखते हैं, B का A के साथ है 1, B का B के साथ होगा 0, ओके B का C के साथ है 1, B का D के साथ है 1 and B का E के साथ है 1। Now इसमें if you see there is no issues, not at all, because your B is connecting with all the nodes, okay so there is no problem in that। अब C का देखते हैं, ओके C का है A के साथ 1, B के साथ है 1, ओके C का C के साथ ऑब्वियसली 0 होगा, D के साथ how far is it 1 2, so that much it's far, so it is 2। E के साथ 1 and 2, that's why 2, okay D के साथ same thing, D का now with A के साथ it is 1 2, समझा क्या, इसलिए 2। if it's with B just one thing 1, with C it is again 1 2 है ना, B DB BC, so 2, C के साथ 2, D का खुद के साथ होगा 0 एंड E के साथ होगा 2 बिकॉज DBBE, समझ में आ रहा है तुमको, तो 2 1 2, ओके 2 1 2 0 2। E का देखते हैं नाउ, E का थोड़ा हर जगह पे 2 आएगा क्योंकि E का A के साथ है EBBA, सो 1 1 दैट इज 2। E का B के साथ डायरेक्ट कनेक्शन है तो 1, C के साथ सेम 1 and 2 दैट इज EB and BC, तो 1 + 1 is 2। D के साथ होगा इसका वापस 1 and 2 एंड E का E के खुद के साथ होगा 0। सो होपफुली यू अंडरस्टूड। एक छोटा सा इसमें कर लेते हैं, अगर सपोज अगर सपोज एक F नाम की कोई चीज होती थी और E का F के साथ होता, सो इमेजिन दिस इज अ नोड, एक एज है और एक यहाँ पे F है, ओके सो अगर A का मुझे F के साथ कनेक्शन होता था ना, सो इट वुड बी 1 2 and 3 बिकॉज वन मोर नोड है। होप यू अंडरस्टूड द फंडा। अगर वो फंडा समझ में आया 1 2 3, तो मतलब ये बाकी सब समझ में आएगा, ठीक है ओके। सो ये एडजेंसी मैट्रिक्स अपना हो गया फॉर दिस थिंग। नाउ ओके, तो नाउ सिंपल ऐड कर लो, 2 + 2 + 1 + 1 6, इसका होगा 4, इसका होगा 6, इसके होंगे 7, इसके होंगे 7। Now this what you have obtained my dear friends is called as your farness, okay। अभी वो है ना, वो हमने जो डर लग रहा था, वो जो इक्वेशन है उसको छुपाया था ना हमने, ओके वापस उसको लाते हैं, okay। So this is your equation for closeness centrality, that is C and C, and this value, okay, summation of dij, I and j were these values, okay, that summation of I dij was, this was the farness ka value। have you got the farness value? Yes, so this is your farness value। now equation क्या कहता है हमको, n minus 1, n is 5 because there are 5, so for all of the entire network it will be 5 minus 1, so n minus 1, that is 5 minus 1 upon farness क्या है 6, so for the first case it is 4 upon 6 which is 0.67। in this case it will be 5 minus 1, that is 4 upon farness is 4 equal to 1। ऐसा करके for all of this what we get is your closeness centrality। so what is closeness centrality? It helps us, it is an basically an algorithm or a mathematical formula which tells us how fast the information should be flowing within them। now if you look in all of this, we will see B का सबसे ज्यादा है which is 1, that means the information which will be flowing will be the fastest, okay। obviously it is connected to all the nodes ना, तो फटाफट ट्रांसफर करेगा। बाकी सब में because it is 0.67 तो थोड़ा स्लो लेगा, 0.57 वाले जो है वो और ज्यादा स्लो हो जाएंगे because it is very far, ओके तो क्लोजनेस नहीं है, 1 मतलब ज्यादा क्लोज है, 1 से थोड़ा कम है मतलब क्लोजनेस थोड़ा कम है, तो क्लोजनेस कम है तो ज्यादा टाइम लगेगा, दैट इज आल्सो, इट्स आल्सो कॉल्ड एज फारनेस। क्लोजनेस कम है मतलब फारनेस ज्यादा है, ओके so understood what exactly is closeness centrality? It tells us how the different nodes, it is a formula which tells us how the different nodes, how much the different nodes are close to the central। हाँ अभी तुम पूछेगे सेंट्रलिटी मतलब ये सेंट्रल किधर है, the central is the B, B you can see is connected to most of these things. So B is your central node here. Alright, it's also called as your hub node. Okay, we can call that, but I won't call it a hub node क्योंकि प्रोटीन काफी छोटा है. इतने छोटे से प्रोटीन के लिए हब बोलना इज लाइक कुछ ज्यादा ही भाव देना है उसको. ओके, अगर बड़ा सा नेटवर्क होता देन वी कुड हैव कॉल्ड दिस अ हब, बट इतने छोटे के लिए इतना भाव नहीं देने का. ओके, सो this is closeness centrality. The second is your betweenness centrality. Now betweenness matlab, if you see, this is A B D and A C D. Now ये एक पाथ है और ये एक पाथ है. अब दोनों पाथ मिले हैं इस जगह पे, that is in this node, that is your D. So betweenness is D is a node which is in between both the nodes. Okay, so betweenness centrality is what we call as a communication flow. Okay, आपको I think पता होगा इलेक्ट्रिकल सर्किट में to move it there is something which actually helps the circuit to go on or off. Okay, so वो communication of the flow करने का जो बीच का जो section होता है, okay, the junction where actually if you've seen the electrical circuit, there's a junction point. So this is the junction point. It is the communication flow. So it allows something to go or not. All right. So that means nodes which have high betweenness centrality, okay, they have a good communication path. So in simple words, what I want to say is betweenness centrality helps to the flow between the pathways. So what we are talking about, these metabolic pathways, क्योंकि अभी तक तुम whatever you're looking at these examples, you are saying, you must be thinking, अरे ये सब क्या undirected network के लिए होता है क्या? Okay, directed के लिए doesn't it work? ये weighted के लिए does not work? No. This I'm talking about. These are all the topological parameters. These topological parameters are a part of your all the networks. They are implemented in your undirected graph network. They are implemented in your directed graph network. They are also implemented in your weighted graph network. Okay, these are the finer parameters. Topological parameters, these are finer parameters which gives us a much clearer picture, okay, of the network. Okay, इसके पहले जो तुमने देखा मैट्रिक्स जनरेट करना, those are all networks, but sometimes you need to have a much finer picture, okay, much better detailing, okay, and that better detailing is nothing but these topological parameters. They give you much better insight, a much better look into how the network is, and why you need to have a much better look in order to analyze it better. Okay, so betweenness centrality tells us that which are the important proteins which are there in the signaling pathways, या ऐसे कौन से drug ऐसे कौन से targets है, protein targets है which can be useful for your drug discovery. Why? Because it is at the junction, वो common pathway, it is in between, okay, the central point. So this was one of the topological parameter, okay, which was your centrality analysis, okay, in which we saw two subparts of your centrality analysis. The next one, okay, is your clustering analysis. Hopefully you understood what is centrality analysis, how it works. Centrality I'll again okay, move ahead. Centrality I'll again tell you. Centrality matlab which is that central hub region, and how the different nodes are connected to that hub region. Based on that, we have two of them. We have closeness centrality, okay, to determine how close things are to the central pointers, and the betweenness मतलब जो network flow होता है उसके बीच में जो communication के लिए help करता है, या this important word you can add, it can control the information flow. So anything which controls the information flow is what is seen in the betweenness centrality. Okay, so these are the 2 points. Now we move to the last point, which is your cluster, the last topological analysis, which is your clustering analysis. Now as the name suggests, cluster मतलब community. Okay, आप बोलोगे सर ट्रांजिटिविटी भी तो क्लस्टर था ना. So ट्रांजिटिविटी भी एक क्लस्टर का एनालिसिस होता है. That is also a property. That is why it is a property of protein-protein interaction analysis. Okay, clustering analysis मतलब हम उसी को ज़्यादा अभी, these are the various when we are talking about cluster, okay, there are various types of clusters. So this clustering analysis will tell me, I mean these few things will tell me what are the various clusters which we are talking about. Okay, so if you see this, for example, this is one network, this is one network, तो ये cluster हुआ, ये cluster हुआ, ये cluster हुआ. इस cluster को हम कहते हैं community. ओके इस क्लस्टर को कहते हैं हम कम्युनिटी. कम्युनिटी इज नथिंग बट दे आर ग्रुप ऑफ नोड्स ओके विच आर कनेक्टेड टू ईच अदर and then they themselves join a rest of the network. So if you remember the properties of networks mein, whatever those small networks what we saw, they were all communities. Okay, so there was one community, other community. So this is what is called as community. Very simple. Okay, by the way clustering analysis के जो भी हम terms या जो अंदर के देखेंगे ना, they're all pretty simple. Okay, so this is community. मॉड्यूल्स. मॉड्यूल्स क्या होता है? मॉड्यूल्स होता है योर फंक्शनल यूनिट ऑफ द नोड्स. मतलब whatever your these nodes which we are talking about, okay, these nodes, or in this case if you are talking about protein protein interactions, they will have nodes, okay, which will not be directly interacting, but they will have but they are very important functional units. Okay, I'll repeat again. They do not have any interaction in the same space and time, so they do not directly take part in the interaction. I mean, they are there, they are interacted, but staying back, they play a very important role in the functional unit when it comes to the protein. So those are what we call as modules. In short, samajh lo, it's basically a node with a functional value. That's it. Okay, that is a module. The next is your complex. Okay, so this is what a protein complex we can say. Protein complex is nothing but a group of proteins which interact with each other. All right, and they are in the same time and space. When I'm talking about same time and space, matlab, it is in that same region. Okay, in the same region, if they are interacting with each other, okay, to form a very stable protein complex. Acha, if you know a little bit of proteomics, you will understand that when we are talking about a stable protein complex, we are talking about that means all the amino acids which are there in the protein are very much nicely interacting with each other. They do not have any steric hindrances with each other. Now this was hardcore protein का फंडा. इसमें ये हम कॉम्प्लेक्स बात कर रहे हैं नेटवर्क के संदर्भ में. ओके, सो नेटवर्क के संदर्भ में प्रोटीन कॉम्प्लेक्स व्हाट इट एक्चुअली मींस इज इट मींस यू हैव दिस ग्रुप ऑफ प्रोटीन्स और ग्रुप ऑफ नोड्स व्हिच आर इंटरैक्टिंग विद ईच अदर, व्हिच मेक्स इट अ वेरी स्टेबल network. Okay, and this stable network, okay, is called as a complex. Okay, that cluster is what we call as a complex. So understood what is complex? So you understood what is community, you understood what is a module, you understood what is a complex. Then the last two terms are your cliques and motifs. Clique, if you look at this diagram, okay, can you see this diagram? You can see node. So you can see 5 nodes, and if you see all of these nodes, each and every node is connected to each and every member to each other. Okay, if you see this entire thing, all of them are somehow connected to each other. ऐसा कोई hanging नहीं है. There is no. If you visualize the other, or if you can go back also on the video, जो इनिशियल हमने प्रॉपर्टीज की बात की थी, ओके, जो एक नेटवर्क बताया था, यू वुड सी सम ऑफ द नोड्स आर जस्ट हैंगिंग और दे आर जस्ट अटैच्ड टू द मेन हब. ओके, वैसा cliques are not that. Cliques matlab all the nodes, okay, are connected, are well connected to each other, to each and every member. Okay, for example ये वाला अगर तुम देखोगे ना, okay, या ये वाला देखोगे, ये हर एक दूसरे के साथ connected है. Okay, so that's why this thing becomes what we call as a clique. Alright, so understood as a clique where all your nodes, each and every node is connected to each and every member. Motif is something like this. Motif is nothing but a subgraph. Okay, it's a very basic term. Motif का मतलब subgraph होता है. That is a small section of the entire network. Okay, so these are also one more thing is motifs, if you remember in your proteomics point of view, is nothing but a small functional subset of a protein. It's similarly in this case also motif is a small subgraph which is there in the network, which plays a very critical role in the protein interaction network. So जो जैसे motif role is there in your proteins, the same motif role plays in your protein interaction network. See, try and understand, these are just terms which are used. Okay, otherwise the terms mean the same thing, okay, in all the cases. Yeah, so if we actually look at the entire protein interaction network, what we did, we first saw what was protein interaction network. We understood what is systems biology. We also understood what is an interactome. Okay, then how are the different proteins interacting with each other? If they interact in a systematic manner is what we call as your systems biology. So when we are talking about systems biology, we have to talk about graph theory. Okay, we saw wonderful history of graph theory, of how one great scientist, okay, for fun he actually started doing something and he came up with a completely new field of mathematics, okay, Euler, okay, which is called as graph theory. We saw the various types of the network edges which are there. We saw three different types of network edges: your undirected, directed, and weighted. Then we carry out adjacency matrix for them. Okay, at the different adjacency matrix. So how are the degrees formed? Usme we learned a very new term or a very new technique. I think that was the first term which we learned in this whole. In this whole journey of ours of PPIN, that which is what we call as degree. Okay, so you understood what a degree was in PPIN, or protein-protein interaction network. I'll start calling it PPIN. Okay, we'll see a time. Next is types of biological network. So yeah, this was in the first two videos. Then in the third video we saw what was your types of biological network. Types of biological network. So that was in the second video itself. Okay, so second video itself we saw what was the I'm sorry, it takes, I shoot it in such period of time, so I sometimes forget. No problem. Okay, so types of biological network and adjacency matrix we saw in the second video itself. So biological network, we saw what is a protein-protein interaction network, how a gene interaction network. Mostly what was important, the metabolic pathway network. What we saw in that was how the different networks are, how they are connected to each other, and what do they actually suggest. Matlab, where is a straight line, where there is a dots, okay, all of these are taken into consideration when we're talking about adjacency matrix. Okay. Then in third video we saw what was your properties of PPI. So we saw three important properties of PPI, that is your small world effect, scale, network, and transitivity. Small world effect is nothing but the closest connection between the two networks. Scale free is how various other nodes which are there, they come together and they are a part of they become a part of this hub protein. Okay, preferential attachment. And transitivity is nothing but small clusters, and how these clusters help us to find out biological analysis. Okay, and in this video, that is our last video, we saw what is topological PPI analysis, in which we saw the two important analysis, that is centrality analysis and topological clustering. So this, my dear friends, was protein-protein interaction network. Okay, a session of protein-protein interaction network. We went in depth of how PPINs or protein-protein interaction network work. Hopefully you have understood it. Okay, and we will come next time with another session. Okay, till that time, bye bye, take care, and good day. See ya.